So we are live on YouTube, and I will allow people into the webinar now. Okay, I'll begin with the housekeeping instructions. Um, mm, uh, yes, so welcome everyone to our webinar. Thank you for coming at Vijayadashmi. Raj, just confirming, can you hear my voice?
Yes, I can.
Okay, great. So welcome everyone to our webinar. These are just a few instructions about audio and chat box and Q&A. This is what you will see if you've logged into the webinar as an attendee. You can, at the bottom left-hand corner, there are the audio settings. You can click on the audio settings. This window will pop up. Please check your speaker and microphone level. So when we unmute you, we will be able to, you'll be able to speak clearly and we can hear you properly. After that, you can just click on the raise hand icon whenever you want to have a question. We encourage lots of people to ask questions. So if you have questions, please raise your hand. Uh, Apart from that, you can also, once you've clicked, the hand icon will turn green. And once you click it again, it will go down again. So once you've asked your question, please make sure to lower your hand, so to speak. The next bit is the Q&A box. You can click on the Q&A box. This window will pop up. You can write your question here. And once you've written your question in this area, you can click send. And you can also tick the send anonymously box here if you do not want anyone to know that you asked a question. Uh, Apart from that, that's pretty much it. Chat is usually disabled for all the attendees. So whatever issues you have, please put them into the Q&A box. This talk is also being live streamed on YouTube. Uh, so apart from that, if you are on YouTube and you have any questions, please just type them into the comment section there, and we will relay them into the webinar. Apart from that, the last bit is that we do not issue any certificates for this webinar whatsoever. This is an endeavor purely for the love of science. So please do not ask us for certificate. Thank you so much. And I will hand it over to Mahin Ali Khan, who's the head of communications, and she will explain a little bit about about our concept of our public lecture series. Over to you, Mahin.
Hello, thank you very much.
And hi, everyone, and welcome to yet another edition of The Human Body Inside Out. This is a communications office endeavor that we present as part of our Science in the City program. And our Science in the City program essentially caters to bringing the science that happens in our cluster to the people outside of the cluster. And it's a way of bridging science and the public. So, this is one of our endeavors, and I'm really happy to see all of you here um, to, to be part of this. I know it's at the end of the week. I know we're on a festival weekend, and everyone's really keen to kind of get off the call and start celebrating with good reason. So I'm not going to take too much time of yours. I'm really happy that we have Dr. Indumati here with us today, but I'm going to let Raj tell you a little more about that. Thanks again.
Just before you begin, Raj, people are asking, is it okay to ask questions in the middle? If you have questions at any point of time, just put them in the Q&A box, please. And if you do really want to ask a question, just raise your hand. And if there is a gap in the conversation, Raj and we will notice it, and we will definitely unmute you so you can ask your question. Thank you so much. Back to you, Raj.
Okay, thank you, Chandrakant. Thank you, Maheen. So it's my absolute pleasure to have invited Dr. Indumati Maripan to give this talk. She's had extensive training in cell biology, stem cells, and regenerative medicine, and she's currently a scientist working at the Center for Ocular Regeneration at the LV Prasad Eye Institute in Hyderabad, probably the top ocular research institute in the whole of India. So today she's going to talk about how the eye works and why do some people go blind. So Dr. Indumati, thank you very much for joining us, and I'll hand it over to you. Thank you. Sorry, you may need to unmute.
Unmute yourself, Dr. Indu.
Yeah. So thank you so much for the invitation and for giving me an opportunity to talk as part of this lecture series. It's a great endeavor, and it's a challenge. It's a pleasure as well a challenge to communicate the science we do in simple terms to common public. So it's a great initiative, and it's my pleasure to talk about eye today. So should I be sharing the screen now?
Yes, please do.
Can you see it?
Yes, we can.
Okay. So very good evening, all of you. Today, let's talk about how our eyes function and why some of us have problems sometimes. Okay, so now this is the how eye looks from the outside. This is like the textbook information that most of us know about. So you have cornea in the front, which is totally avascular and transparent structure, which can be appreciated in the cross-sectional view here. And behind that is what is appearing as your dark circle is nothing but the iris, which is nothing but which acts very similar to the diaphragm in a camera, which regulates the amount of light that enters the eye. And this iris creates an opening called the pupil, which is a window through which the light enters the eye. And the light entering the eye hits the lens, and the lens helps in focusing the light back to the retina, which is a screen at the back, which captures the light signal and converts into electrical signal and passes it on to the brain via the optic nerve. So this is broadly the anatomy of an eye, and you have two major chambers. One is the in the front filled with aqueous humor, which is more of a liquid, flowing continuously secreted and drained liquid. Whereas vitreous chamber, it's a very viscous fluid. Together, it gives the shape for the globe. It helps in maintaining the globular structure. So now, before we go on to how an eye functions, let's also look at the embryology, how an eye develops. So eye is nothing but an extension of the brain. So here you can see that as your brain develops, this is the wall of the forebrain. And there are extensions coming out of the forebrain. This is a neuroepithelium, which is derived from the neuroectoderm. And you have an optic vesicle that is formed. This optic neuroectoderm is surrounded by the ocular surface ectoderm. And this surface ectoderm also extends. So when the neural tube forms, the neuroectoderm merges and you get a continuous epithelium of neuroepithelium and the ocular surface ectoderm as two contiguous epithelial structures. So in development, a lot of organs are formed just by evagination of cells, proliferation, evagination, and invagination resulting in formation of different organs. So here you can see that the eye structure is formed by evagination to form optic vesicles on either side of the brain, of the axis, central axis, which goes on to touch the ocular surface ectoderm. And the region where it touches, it gets the signaling cues from the ocular surface ectoderm and then starts invaginating. It's going inwards now to form, resulting in the formation of a cup-like structure. So this neuroectoderm that invaginates inside now forms the future retina, and the outside forms the ocular surface, your sclera, and the RPE develops here. Similarly, the ocular surface ectoderm also invaginates, and this forms a vesicular structure inside, which then develops into a future lens, and completes the structure with the ocular surface ectoderm, remaining ectoderm, contributing to the formation of cornea. So this pictorial representation nicely explains what happens during development. There's an initial evagination to form the vesicle, and then clubbed with the invagination resulting in the formation of retina and the lens-like structures, together contributing to the complete organ formation. So all this takes place within 8 weeks of gestation. So here is the line diagram that shows how the light enters the eye, and here is the leaf, and the light coming from the leaf enters the cornea, and then the lens, and then hits the retina where it is perceived. So the refraction of the eye is mostly dependent on the lens and cornea, mainly cornea. 60% of the refraction is contributed by the cornea. So if there are any problems in the cornea, there is a severe, the amount of light that enters the eye is severely affected, and therefore the vision is affected. And the remaining 40% contribute to the lens. We generally think that lens is the major thing that contributes to the refraction, but actually it's the cornea that contributes to the refraction. And finally, retina helps in image perception. So we'll go one after the other and see how each of these organs within the eye functions and helps us to perceive what we see. From the retina, the electrical impulses generated by the photons are then transmitted to the brain via the optic nerve, and then it reaches the visual cortex, which helps in visual perception. I'm not going to the detail of this part aspect of it, as Bhavna in one of her earlier lecture series on brain has explained this in very good amount of details. So I'll spare those details for this talk today. So many of us would have visited an ophthalmologist at some point of our life, and we would have, the moment we go in, and we'll be asked to look at this, some kind of a chart like this, and we'll be asked to read which line, till which line we can read perfectly. So here the numbers represent, this is a Snell chart. Here the numbers represent what person's visual acuity is. This is in terms of feet. A person with a 20/70 vision means A normal person can see something at 70 feet. A person with a visual impairment can see only if he comes closer at 20 feet. That's what it implies. So 20/20 is a perfect vision. And if you have anything above, that means you have some visual problem and you need glasses for refraction or any other intervention. There are some people who have very above normal vision, who can see even farther. And there is also another chart called Ishihara chart. So this is also something some of you might have noticed. This helps in detecting any problems in color perception. So there is also black and white perception or grayscale perception. And you have color vision where there are different types of cones that perceive the light of different wavelength, like your blue cones, red and green cones. So here this chart detects your green and the functions of your green and red cones. So this helps the ophthalmologist to figure out whether you really have any problem with visual acuity. So it's not to do with the total perception, but it's only to do with the visual acuity. So the refractive errors can be of different types, together contributed both by the cornea and the lens. Here, in case of myopia, you have the image is not formed on the retina, but in front of the retina. So you need to be this need to be corrected by having a convex lens so that the image is focused onto the retina perfectly. As in case of hyperopia, it is projected beyond the retina and needs a convex lens for focusing onto the retina. There's also another condition called astigmatism. Here, the problem is not in the lens, but in the cornea, shape of the cornea. Here, because of different thickness, we'll see the details of the cornea later, but just take my point that the cornea can have different thicknesses all across, and this can cause astigmatism, and this will also have a serious effect on your light refraction and need to be corrected. So these are basically some of the basics or real life experiences many of us would already have. So other than that, the aged people might experience this problem, presbyopia. This is an age-related ciliary muscle problem. Here, it's not the problem of the cornea, but it's a lens accommodation issues. Here, this is an example where you can see that when you're seeing light coming from far off, your lens is more stretched out, whereas when you have to focus on something that is closer, your lens have to be constricted. This is mediated by the muscles that are attached to the lens. And if there is any problem in this accommodation or the movement of the muscles, then you have issues in focusing on objects. Mainly the closer objects, like when you want to read something, small prints, then it becomes a problem. And you cannot accommodate, and then we simply compensate by just moving the object a little farther. So this requires reading glasses. This is another something that many of us would be aware of. So another condition called keratoconus, where the corneal strength is not so good that it assumes a conical shape. Here, not just the refraction, you also have to maintain the refractive corrections. You have to have a proper structure to hold it. In this case, people recommend, the ophthalmologist might recommend wearing a contact lens to hold the cornea in shape and also to maintain the refractive corrections that they prescribe. I also have put up this slide because whenever we go to the clinic, they apply some drops onto the eye and we wonder what the drops is for. So if you have to examine the structures at the back of the eye, you need to reach, the light has to reach to the entire retina at the back, for which your pupil has to open. So there are drugs that are given, the drops are nothing but dilators. It dilates the muscles, relaxes the muscle, so that your pupil, which is a muscle structure, it relaxes and opens up. So the iris muscles relaxes, and that leaves a large pupil opening for more amount of light to go in, so that imaging of the retina becomes very clear. So this is what happens in a dilated eye. So when you're in bright light, this will be the situation. When you're in a dark situation, your pupil will be totally dilated. So this is important to know later, so about the functioning of the iris. So now let's move on to understand more details about how each of these tissues are made of and how they contribute to the vision and what happens when something goes wrong. So cornea, let's start with cornea. So cornea is the front part. As I said, it's shown here. It's not just a simple structure, but it's a very complex structure made of different cell types. We'll look into that in detail. So it's a very transparent structure, as I said. But the cornea, every time we blink, the cornea has thick epithelium on the top and a thick, about 100, 150 micron in thickness. It's a stratified squamous epithelium, just like our skin. Yeah, it has stem cells at the base, and it can help in regeneration. Every time you blink, there'll be a lot of cells that are shedding on a day-to-day basis, and these are regenerated. It is believed that in a year, complete corneal surface is replaced with fresh set of cells by homeostasis. So you have a thick stroma, which is about 600 to 700 micron, made of collagens and keratocytes. It's a kind of stromal fibroblast cells that makes these collagens. The unique thing about this is your collagen fibrils are nicely organized in compact structures, because you can't afford to have irregular arrangements, because that will affect the light entering and the refraction of the light. So nature has arranged, made in such a way that the collagen fibrils are tightly packed and arranged in a linear fashion, and helps in maintaining the proper transparency of the cornea. There's also, and cornea is avascular, that means there's no nourishment for the cells on the epithelium. So somehow it has to be nourished. This is taken care of by the endothelium at the behind. So these are specialized cells at the behind. Hence, they have expressed a lot of fluid pumps and proteins that helps in transporting the ions and water across the tissue. So if there is any problem in that layer, third layer, then this water balance in the cornea is affected, and a lot of water gets retained in the stroma, collagen swells up, and then you have a hazy cornea or edematous cornea, that means swelled cornea, which will affect again the refraction. So each of these layer has a unique function, and also the water that is entering here helps in nourishing the cornea, because there's no vasculature in the cornea. You can't have a vasculature because that can also refract the light. So that's the unique structure of the cornea. So the stem cells for the cornea resides not in the center, because stem cells need nourishment. You can't have stem cells in the center because there are no vessels. They're conveniently placed at the periphery or 360 degrees in a region about 2 mm in diameter. In limbal crypts, these are unique structures called limbus, where the stem cells resides in the basal epithelium, and it regenerates by dividing, proliferating, and migrating in a centripetal manner, peripheral to central, and then from center it's basal to apical migration, just like how it happens in epithelium. So that's the difference of corneal epithelium and compared to skin epithelium. So this is in detail. As I said, it has a thick layer of epithelium. The basal cells are the proliferating TA cells. You have a Bowman's, thick Bowman's, which is the basement for the epithelium. Thick stroma with collagen fibrils and stromal keratocytes, which keep secreting this and maintaining the stroma. And the endothelium, which has the pump functions and the thick decimus membrane, together contributes to the corneal structure and the function. So here is a condition called FECD, Fuchs endothelial dystrophy. Here, the endothelium is a nice monolayer of cells, nicely arranged cobblestones phenotype. They are neural crust derived cells, and they undergo degeneration for various conditions. Here it is genetic disorder in a particular gene. And because of that, the cells die, and these cells do not regenerate just like corneal epithelium. Corneal epithelium has limbal stem cells to regenerate, whereas endothelium never regenerates in adults. So any cell loss accumulates, cells leaves gaps in the endothelium, and they expand to fill the gaps, but then the cell density goes down as the condition worsens, and finally cornea becomes opaque. This kind of structure, you can see that you have guttae formation. Wherever the guttae is there, cornea becomes edematous and it's opaque, and it's a cloudy cornea. So this is what happens. How do we treat this condition? So either the corneal epithelium or stromal problems or endothelial problems can be corrected by a simple corneal grafting or the corneal transplantations, which is well known. This is the only organ transplant that doesn't require immune suppression because there's no vasculatures. So how it is done? So the entire cornea, so the problem is in the central cornea, the button is removed and the donor cornea is taken, and the central cornea is being sutured. So here is a donor cornea, people who have donated their eyes. The only part of the eye that is donated that's been used in transplantation is the cornea. So here they don't take the limbal region because it has vasculatures that will cause problem in immune recognition. So only the central corneal button is taken and this is transplanted. It's like sutured. So this gets integrated and now the person regains the vision. So this is a simple treatment for some of the corneal surface problems, either in the corneal epithelium, stroma, or endothelium. So, but there are some conditions where there are extensive damage such as burns or acid-alkali injuries, wherein the entire corneal surface is damaged. Here is a nice cornea with the limbal regions marked here. Once the limbal region is also damaged, there is a severe damage. The conjunctiva, which is the white part of it, which is also a contiguous epithelium, starts migrating onto the cornea. And it not just the epithelium comes in, but also the vasculatures comes in. So it now, the immediate response is to heal the wound. Wound is healed, but then the corneal surface transparency is lost, and therefore the person cannot see. So this is a problem called limbal stem cell deficiency. Here the corneal transplantation will not work because your vasculatures are there and your allogenic grafts will be immediately recognized as non-self and will be rejected. So the only treatment option in this case is to regain the limbal barrier, because this not only acts as a niche for the stem cell for epithelium, it also has some signaling which prevents the conjunctival cells to come in. So this needs to be ensured. So this is done by limbal stem cell transplantation, wherein a small biopsy is taken from a normal healthy eye. So it's an advantage. You have a fellow eye where you can harvest the cells, and the stem cells are residing in a particular region which can be harvested, taken, and then you grow them in the lab as a sheet of epithelium. And this sheet of epithelium, so you can see an expand culture where a nice growth of epithelial monolayer is established, and this can go on to the patient. Here it's a severe burn injury patient with severely damaged eyes, total LSED, and after treatment, you can see that your corneal surface clarity is totally regained. So the success rate of autologous stem cell transplantation, limbal stem cell transplantation, is about 70%, 75% at the end of 1 year. We have been doing this at LV Prasad for past 15 years, started in 2002. Now over 1,000 cases have been treated, and the longer term follow-up, we can have approximately about 70% success rate. The 30% where it doesn't succeed is because not here we are transplanting only the corneal epithelium. We are not dealing with the stroma or the endothelium. The problem is deep in the stroma, then just reconstructing the corneal surface epithelium will not help. So the patient but has an option to go ahead with the corneal transplant because now the corneal surface is patent. No vasculatures are there. So now moving on from the cornea, so the cornea not just receives nourishment from the aqueous humor behind, mediated by the corneal endothelium. The surface fluid, which is the tear that is coming onto the surface, also helps in nourishing and keeping the ocular surface wet. So if it is dry, then you have a lot of other issues such as pain and sand or foreign body sensation, and so many other complications happens. So this is mediated by the secretions coming from few glands, not just one gland. Lacrimal gland, we know many of us, which is well known. So it secretes the tear or the liquid part of it. There are two other cell types called the meibomian glands, which are there all across your lids. So these lids, glands drain at the edges of the lids, and they secrete the oil layer because you need a layer on the top to prevent the evaporation of the fluid. So that forms a top layer. And there are also glands goblet cells that are present in the conjunctival epithelium secretes mucins, which helps in even spreading. Basically, it allows the fluid to evenly spread all over the corneal surface. So there are three layers: mucin layer, liquid, which is the lacrimal secretions, and then you have the fat secretions from the meibomian glands. Together forms the tear fluid on the surface, which helps in nourishing and keeping the ocular surface wet. So if there is any problem, then you have problems of dry eye syndrome, which is a looming problem of late, with a lot of people sitting before the computer, and we'll talk about that little later. This is what I was telling about your meibomian glands along the lid margin. So they drain their secretions at the edge of your lids. You have your lacrimal gland secretions. Together helps in maintaining the corneal surface integrity. So there are also this lacrimal drainage problems. So because whatever the fluid that is secreted, it has to drain out somewhere. So this is a normal homeostasis. Whenever we cry, the excess fluid comes out. We shed the tears. But during regular homeostasis, also a lot of secretions happens, and the drainage happens through the lacrimal, nasolacrimal duct into the nasal cavity, and this drainage doesn't happen or gets blocked in some people, that causes fluid accumulation on the surface of the eye. That can also affect your light perception. So other than the surface, the interior of the cornea, that is anterior chamber, is filled, as I said, filled with aqueous humor, which is a fluid which is constantly secreted and drained, which helps in the nourishment of corneal endothelium, corneal stroma, and the epithelium. So this secretion comes from the ciliary muscle. Muscle has a lining of epithelium. These epithelial cells secretes the aqueous humor, which comes in from behind, from the top of the lens, and then drains out, goes into the anterior chamber, and then drains and at the margin. This is the angle between the cornea and the iris. So there are certain structures called trabecular meshwork cells, which are nice meshwork-like structure. Basically, it allows fluid to go out. There is a drainage pathway there. So if there is any problem, whether it is congenital because of this angle problem, angle formation developmental problem, or because of any other conditions which affects your trabecular meshwork, then the drainage becomes a problem. So what happens if the fluid is not drained? Because it's constantly being produced but not drained. So therefore there is a lot of fluid builds here, and that creates a pressure. So that's what causes glaucoma. So you have too much fluid. That fluid causes the pressure, puts it on the, transmits onto the lens back to the retina, onto the vitreous cavity. Now your retina is constantly under pressure, and then photoreceptor cells at the back suffers severe damage and loss of vision, apart from other problems such as nausea, your pressure, or your headache, and all other issues. So these these are very severe vision threatening problems that can be dealt with not by meddling with the physiology, but in simply finding a way to drain the fluid. So here, Ahmed, the glaucoma valves can be implanted, wherein you have a drainage linked directly to your anterior chamber, which just drains the fluid outside. So this helps in relieving the pressure, and therefore your constant chronic problems on the retina is averted. So that's what a glaucoma treatment or a glaucoma surgery involves. Now, coming to the lens, which is the next refractive unit. Here, lens, as I said, it forms from the ocular surface ectoderm because of the invagination. So this invagination initially forms a nice circular structure, which is basically a vesicle, a monolayer of lens epithelial cells with the lens vesicle in the center. And the lens vesicle and the epithelial cells at the posterior end close to the retina starts differentiating in the sense they become like fiber cells, they're elongated, and the nucleus, as they mature, is lost. They lose their nuclei, and resulting in the formation of nice lens fiber center core. And this is important because you're, again, just like a cornea, you can't have any other reflecting surfaces inside the lens that will affect light from entering. So you need a clear entry. So this, again, unique anatomical plan has enabled transparency for the cornea, sorry, for the lens. So this is what I was telling about the lens fiber cells. And here in a mature adult adult lens, you don't see any nucleus in the fiber cells. It's totally anuclear. This is the H&E image of how a developing lens will look like. So what happens if there is any problem? So in cataract, again, many of us would have known cataract surgery because the lens becomes cloudy. So earlier it was cornea becoming cloudy, here it's lens. Lens can become cloudy for various reasons. It can be age related, many of us know. So old adults of more than 60 can have this problem. Lens is made of mainly the proteins called crystallins, and these crystallins can undergo some changes, and that leads to cloudiness. So the cloudiness can be of different extents, can be in the center, can be in the periphery, or the entire lens can be can lose its transparency, and therefore your light entering into the eye will greatly be affected. So what is done in case of cataract surgeries is nothing but, so you use a laser. It's a phacoemulsification when you go in and then you basically mush up the entire content inside and suck it out, leaving the lens sac intact. Inside the lens sac, once it is clear, yeah, intraocular lens, which is a synthetic lens, is inserted, and this keeps the refraction. So this helps in focusing the light to the retina. So this is what is being done in case of cataract surgery, which is now a very routinely done practice, which is completed in less than half an hour. So now coming back to the back side of the eye, which is the retina. So you have, again, retina is a very complex structure. As I told you earlier, it is derived from the neuroectoderm as an extension of the brain. So majorly made of neuron cells, different types of neurons. And it's made of 3 different layers. Outside is a photoreceptor layer where the actual cells that picks up the light signal or the photons entering the eye. And then you have interneurons which transmit the signal from photoreceptors downward. And then finally your RGC layer, which is the retinal ganglion cells, which has long axons which takes a signal from the retina to the brain via the optic stalk. So we'll look that into details. You have photoreceptor cells, which is the outer nuclear layer. You can see the 3 different layers of cells. Outer nuclear layer, mostly your photoreceptors. And in human retina, it's rod-rich retina. So most of the nuclei is rod, and you have cone cells scattered. And inner nuclear layer made of 3 different neurons and glia. And then you have RGCs, which finally converge together and exit the retina and reaches the brain. So different cell types, as I said, each of them have unique proteins to express as markers, and that can be used to identify any particular problem. Here I've shown this is the rhodopsin staining, nicely marking all the rods in the outer nuclear layer. And here it's blue cones. As I said, human retina is rod-rich retina. Rods are more, cones are less. And cones, here is a blue cone which is scattered here and there, and your interneurons express your calvine, horizontal and Macarene cells, bipolar cells, nicely beautifully labeled by PKCs and CHX10, and your glutamine synthase marks your Mueller glia, the only glial cell type that is present in the retina. So the most important cell type is the photoreceptors. We all know that it's a textbook knowledge. So they have a unique structure called the outer segments, which are nothing but an extension of the cilia. They're modified primary cilia. So their membranes are nicely elaborated and invaginated to form disc-like structures. And right inside the disc is where the photopigments reside, and they capture the light signals. The purpose being, again, to increase the surface area. So the more number of photopigments can be accommodated per cell. And photoreceptors never work in isolation. They also need a particular, another cell type, which is also neuroectoderm derived cell type, but it is an epithelium, not a neuron. This is retinal pigmented epithelium. This is at the back end, which is pigmented. So it acts as a light filter. It captures all the excess light that exists in the retina. It doesn't allow it to come back. And it has unique extensions, and photoreceptors and RPE play a major role in visual transductions. There's a visual cycle, a lot of intermediates that are the chemical reaction that goes on, and some of the intermediates goes on from the photoreceptor to RPE for recycling, and then it comes back. So it works in as a cyclic, as a combined unit together, and helps in visual processes. So any problem, either in photoreceptors or in RPE cells, can lead to severe visual perception. So here is an LIEM picture, SEM picture, where you can see beautiful rods, rod-rich retina, and here and there you can see the cones, which are morphologically, the outer segments are different as compared to the rods. So coming to phototransduction, again, I'm not going to elaborate this in detail because Bhavana has already talked a bit about how the signal is perceived in the retina. But what I wanted to convey is right inside the disc, you have the visual pigments which capture the light along with, and that triggers a series of signal transduction events involving several proteins. And what it eventually does is there are a lot of pump proteins that allows regulate your ion transport across a membrane. So when there is no light, your the pumps are open, the ions freely move in. But when the light is activated, the pumps are closed, and therefore the ions accumulate. That creates a potential difference across the membrane. This potential difference or the electrical impulse helps in releasing a lot of neurotransmitters. These neurotransmitters are then perceived by the neighboring cells, which is the next cell line of cells in the inner nuclear layer, which amplifies the signal and then sends it to the brain. So since I said there are a lot of proteins that are involved, and some of the intermediates in fact go to the RPE and to get recycled. So any problem in any of these proteins, be it the visual pigment, which is the opsins, or any other intermediate enzymes, it can lead to defects in phototransduction pathway, and therefore light perception cannot happen. That's what happens in many of the retinal degenerative conditions, which are genetic disorders, and many of the mutations in many of these proteins are identified to be the cause for visual problems. So as I said, it can be genetic. It's mostly genetic, except for AMD, age-related macular degeneration, where the RPE cells degenerate with age. But most of the retinal degenerative conditions are genetic, inherited. And so if the parent has a mutation, then the mutation can be transmitted to the progenies, depending on whether it's dominant inheritance or recessive inheritance. The severity of the disease changes. And so here is an example in case for non-science audience, mutation is nothing but a change in the genetic code. So genetic code, we have single letter codes. DNA is made of nucleic acids, and you have each each codon is represented by 3 letters together. This is the central dogma. So wherein the DNA is converted to RNA, RNA is converted to protein. A single unit is comprised of 3 units of DNA. You can consider it as a building block or your bricks in a building. So three different combinations together codes for a particular amino acid. A series of amino acids together make the protein. So for a complete protein to be functional, so you need the proper code to be translated. So if there is a problem, here is an example. You have TGT, which is coding for cysteine amino acid. It's now converted wherein the T is mutated to A, wherein, so this code doesn't code for any amino acid, which is a nonsense mutation. We call this as a nonsense mutation, wherein a particular code doesn't code for amino acid. Therefore, there's no amino acid added and the protein is terminated. So you don't get the full protein made. So therefore, because of the truncated or a non-functional protein, you have problem in the function. So this is what happens in many disease conditions. So what happens in case of a genetic mutation, in case of retinitis pigmentosa? Here, retinitis pigmentosa is a genetic conditions where anywhere, there are congenital conditions also. Congenital to anywhere between people in the age group of 40 to 50 can also be affected. It mainly affects the rod. So it starts from the periphery because the cones are concentrated in the center. So your cones are for bright light vision and color vision. Rods are for dim light vision. So periphery is affected. So you have problems like your night blindness will start. Probably it will be difficult to see in the evening, but your bright light vision will be fine. So this is how a patient with the RP visualizes the world. So what you see normally, you have a tunnel vision, only the center you can see it clearly. Whereas in case of AMD, age-related macular degeneration, so macula is a central part of the retina. So here your degeneration starts in the center, that means your cones are getting degenerated. So your daylight vision and color vision will have a problem, whereas your dim light vision will be fine. So this is how a person with AMD perceives the image. So unfortunately, there's no treatment in this case. So only visual rehabilitation is done, and some nutrient supplements are given to delay degeneration, but no real treatment, because retina do not have any stem cell like corneal endothelium, and retina also do not regenerate. So any loss that happens in adulthood, it accumulates and gradual degeneration, eventually leading to blindness. So only visual rehabilitation is possible. Now people are looking at possible options such as stem cell therapy, gene therapy are being explored. Some are in the early stages of clinical trials, but many are in the very early basic research stage. So since the stem cells are not there in the retina, so you have to look for alternative stem cells. So best source is the pluripotent stem cells, that is your cells that are from the embryo, which is capable of making any cells of the body. So if you want to make retina, it should be able to generate retina, provided you give the right conditions. So this has been widely explored, and now recently in 2000, a decade back, Yamanaka et al. Has shown, this is a Japanese group, have shown that not just embryonic stem cells, even somatic cells like any of our skin cells or blood cells can be made or genetically reprogrammed to become like embryonic, in the sense that they can generate any cells of the body. This is by genetic manipulation, by introducing certain genes, and these cells are called induced pluripotent stem cells. They behave very similar to embryonic stem cells in many ways, and they can reproduce many cell types of the body, regenerate or differentiate into cell types of the body, including eye cells. And many groups around the world, including our group, is focused in this particular area, where we aim to use these cells, stem cells, for understanding retinal biology, and also to figure out a stem cell based therapy for regenerating such diseased retinas. So way back in 2011, earlier, this Japanese group have shown that just like how an embryo forms, like you have two optic vesicles formed on either side of your neuroectoderm, anterior neuroectoderm. So here also in the dish, these cells can form bilateral optic vesicles in the dish. This is happening in the dish. So it is possible that you can have culture conditions where you can completely regenerate the whole eye, just like how it happens. It basically replicates what happens during development in an embryo. And these structures not just form one specific cell types, so all cell types of the retina. You have the outer nuclear layer marked by rhodopsin, I think here, inner nuclear layer cells, and even the RGCs. All three different layers can be formed in this. Here it's a brain and eye organized together in this particular image. So our lab also is working, as I said, in the same area, where we are trying to use this patient-specific models to understand retinal biology, and also to, because it's an eye hospital, we are interested in developing a stem cell-based therapeutics for this particular, for at least some forms of retinal dystrophy. So we were able to establish protocols for developing retinas, this multi-layered retina, as I've shown before, and also RPE cells, because both RP and retina is important, and both undergo degeneration, and it's important to transplant both these cells for effective therapy. So we can make sheets of, these are nicely pigmented cells. You can see that you can purify them, and these cells in lab culture conditions can mature and get pigmented, and uniform mass of cells, which can be used in all transplantation or basic research studies. So we are currently working on animal experiments. So eventually the idea is to suppress, so inject these cells in the subretinal space. Your retina, you have a subretinal space wherein you can reach and inject these cells either in the form of a sheet or suspension, so that they can home in and do the job and help in regenerative processes. So that's a lot of groups around the world are now trying, and a couple of groups have already gone into phase 1-2 clinical trials, which is ongoing. So that's hope for retinal degenerative conditions. So apparently gene therapy is also explored, because it's a genetic disorder. You can have vectors or viruses that can deliver a normal copy. So if there is a mutation in a cell, so because of that mutation, the protein is not expressed. What if you deliver a normal copy of gene into the cell and make the cell produce sufficient protein to regain its function? So that's the idea behind stem cell therapy. And one of the, behind gene therapy. So one of the gene that's been widely targeted is RP65, and you also have a commercial product called Luxturna now already in the market, which is available for RP65 gene therapy. But this is useful only if a patient has mutation in RP65. It is not useful for any patients, but only for patients with retinal LCA, which is a congenital form of retinal dystrophy and has mutation in RP65. It's important to note that at least 60 to 70 different genes were implicated in dystrophy, and this is one of them. And the idea here is the RP65 gene packed viruses is delivered subretinally. And hopefully, if the cells are alive, and they will take up the virus and will start expressing the protein and regain its function. So that's been tried for many other gene also that's in the process. And coming to tumors, there are also tumors that form which can affect the vision and also even can be life-threatening. So one important example is retinoblastoma. Here it is a tumor of the retina. So the developing retina, the progenitors, starts proliferating abnormally and gives rise to tumor. This is a childhood disease very early, within just 1-2 years after birth. Children will have problems in the eye. It literally fills the entire globe, and it can also metastasize to other parts of the body and can be life-threatening too, unless it is attended to at the right time or the eyeball is enucleated and removed. So here you can see in a cross section, whole ball is filled with the tumor, and this is a histology showing the tumor mass. So what is done? Radiation therapy, of course, and also chemo. Chemo is done. See if the the tumor can be regressed, and the eyeballs can be salvaged. If it doesn't happen, then the whole eyeball enucleation is done to prevent metastasis, because the whole idea is to protect, to contain the disease, and also preserve the vision as much as possible. So the other forms of tumors are your surface, your corneal surface, conjunctival epithelium undergoes problems. They are all exposed to the outside light, UV radiations, and they can undergo tumorogenic transformations. And melanocytes of the iris and also behind its choroid also has a lot of melanocytes. They also can give rise to melanoma. Again, chemotherapy or enucleation is the solution for this condition. You also have ocular muscles, which helps in eyeball movement. Your left, right, top, bottom, and then rotational movement, rightward and leftward movement, and different ocular muscles are involved in the process. If there's any problem by interfering with the particular ocular muscle that's involved, this is a surgical procedure again, which helps in tightening these muscles. And it helps, you can see that before and after squint surgery, it can be corrected. So these are some of the problems which can be either surgically corrected or by simple refractive error corrections. So in summary, so the main cause of vision impairment is cataract, and refractive errors simply can be corrected by cataract surgery and by prescribing suitable glasses at the right time. Cataract, it's very important, particularly if it's a congenital, because congenital, again, crystalline mutations can cause congenital form of cataract, and your lens development and vision is very important for retina, because retina develops post-birth also. So if light doesn't enter the retina, even retinal development will be affected. So in congenital cases, it's important to interfere the right moment so that the child regains vision, which allows the retina to develop further. If not intervened at the right time, and even if the correction is done later at adulthood, retina will not function. So therefore, timely intervention is very crucial. And I haven't talked about many of the infectious diseases. Dry eye condition, again, is a looming threat now, with a lot of us spending time in front of the computers and blinking. So take your time to pause and then blink in between, and so that you let your lacrimal secretion spread all over, all over the corneal surface. That is not done, you are risking yourself with dry eye conditions. And myopia is again a major looming threat with all indoor activities and near work activities. Like now, Corona has done a wonderful thing for that. So let's take time to go out and see the outside world, expose ourselves to the bright sunlight so that this problem can be averted. And diabetic retinopathy, again, our lifestyle changes is required, and proper diet control and glucose control is very important to prevent a diabetic retinopathy. Here, your retinal vasculatures will become abnormal and starts leaking and causes severe damage to the retina. So, and late detection is, as I said, many of the conditions like cataract or congenital cataract or even retinoblastoma need to be properly identified and treated at the right moment. So time is very crucial. If that is left, then you can risk vision. At the same time, it's possible to risk the life of the individual also. So I want to leave you with this final message that about, this is a statement by World Health Organization about 2.2 billion people worldwide are visually impaired, out of which 1 billion cases could have been prevented. That means preventable blindness is about 75% and can be avoided. It's important. The awareness is very important. So remaining 25% is we really don't have a proper treatment, and just like I told about retinal dystrophies, a lot of scientists are working while we are trying hard and trying to figure out some solutions for these problems. As informed individuals like us, let's spread the awareness to reduce the burden of avoidable blindness so that all may see. So with that, I end my talk, and it's my pleasure to share some of our experiences and knowledge with all of you guys today. And I'll be happy to take any questions. Thank you.
Thank you so much, Dr. Indumati. So there's a number of questions on the Q&A box. If anyone wants to ask a question, they can raise their hand as well. I may ask various people who have written their question to speak them out. So Neha Folthaka, if you're still here, are you able to do you want to say your question?
Neha, we've unmuted you, so you can ask your question. Varsha is also wants to ask. So Varsha, we've unmuted you as well. You can please.
Okay, Varsha, please.
Varsha, you'll need to unmute yourself.
Okay. Are you able to hear?
Yes.
Yes, yes.
Actually, ma'am, on 2080, my LASIK surgery was done. Now it is completely fine. Still, I can able to see everything. But during the night time, I feel that my eyes are very dry. And as I wake up in the morning, I unable to open my eyes properly. Some pain going to occur on my eyes, or as due to the pain, the liquid automatically come out, and afterward I can open my eyes. If this is happening regularly, so I visited doctor. They give us me the liquid like some kind of gel, they drop they give us, and as I put every day, the little bit relaxation is going to happen. Is this way to cure this kind of things are going to happen in my eyes, or any other solution?
Yeah, as I said, it's clearly a dry eye symptom, and so the tear supplements that your doctor have prescribed is definitely useful. And just as I'm not a clinician, but I'm a researcher, but I can suggest from the experience of seeing so many of the patients. Right after you get up, it's useful to have a hot water pack of washes that will help you to relieve your ocular surface. Okay, so that will be useful because it will help in clearing the lipid that is blocking the drainage system. So once you clear the passage, then the fluid will start moving freely, and you will feel relaxed after that. So that partly helps. Probably you can try that. And if you have severe problems, I think it's better to consult a doctor.
Okay, ma'am. Thank you.
Okay, so I'll ask Neha's question for her. She asked whether limbal stem cell transplantation is widely available.
It is definitely been done for a long time at LVP. A lot of patients have been treated. Now, I've explained a culture-based procedure where you take a biopsy, make a sheet, and then deliver it to the patient. Now it has evolved into a surgical method where it can be transplanted directly onto the patient eye. Now it's called SLET, simple limbal epithelial transplantation. And this technique has been trained to a lot of clinicians, not only in India, but also abroad, and it's been widely practiced now. So if anybody is interested, maybe you can get in touch with me. I can put in touch with the doctor who will be helping out in this process. So it is definitely available in the country and also abroad.
Okay. So Rashmi R, are you still there? If you want to, do you want to say your question? If you want to say out your question, you can. If you don't, I will say it out for you.
Rashmi, you are allowed to speak. You can unmute yourself and talk now.
Okay, maybe Rashmi is shy. So one of the questions Rashmi asks is whether there are treatment options for diabetic retinopathy.
Diabetic retinopathy, there are two types. One is wet, and there is another one called dry retinopathy. So dry. So in the case of wet condition, there is a leakiness of your blood vessels, which can be treated, and there can also be retinal detachments that can happen because of retinal diabetic retinopathy. And in case of blood vessel leakage, you have anti-VEGF therapy. So these are antibody-based therapies that are available, and it's been routinely given in the clinic. So that's the only treatment option that's available. And in case of retinal detachments, there are procedures to reattach retina back to the sclera. So that is a surgical technique that's also done by the clinicians. So anybody has problems should be consulting your retina clinician for a detailed understanding and suggestion.
So Shreyans Jain, do you want to ask your question? Okay. So Shreyans asks a very nice question, which I think is actually, so here, I think he's allowed to talk.
Shreyans, yeah, Shreyans, try to unmute yourself, please.
Hello, can you hear me?
Yeah.
Okay. Thanks, ma'am, for a very nice presentation. I'm a researcher at the Institute Pasteur, and basically I deal with mostly mechanobiology of tissues, and my PhD was in migration, but I was very curious first to know what kind of mechanobiological where there is pressure in the eye. But in the same line, I was very curious and wondering that myopia and hyperopia is like a long-standing problem, and so many people suffer from it. And only intervention, clinical intervention, which is done is the laser surgery, mostly. But I was wondering if corneal or the cells in the lens will show that if you kind of cyclically stress stem cells, then they can proliferate more, or their stemness is more increased and all. So I was very curious to know if there has been any kind of research done, or there are some hypotheses which you can postulate to have a mechanobiological intervention to the tissues consisting in corneal and lens region in order to correct or revert back the conditions like myopia and hyperopia. I can make myself again clear. I might be asking a very naive question, but I was very curious to know from an expert viewpoint, what do you think about it?
So from with the perspective of corneal problems, so you have your epithelial defects and you have ways of dealing with the epithelial problems, including stem cell therapy is already available, right? So in case of stroma, like in case of keratoconus, where your strength of the cornea is so weak because your collagen fibers doesn't hold and it takes up a conical shape, you have a problem there because of certain gene mutations, which are important for holding these cells together. So that's a genetic conditions. Again, you cannot do anything more. But then there are UV cross-linking procedures that are done to cross-link these proteins and hold them together. But then I don't see it as a permanent therapy because it can only give a temporary fix. Similarly, for endothelial keratoplasty is done, or your full corneal thickness transplant is done. So that works perfectly fine for even in, because FAX or CHED or many of these endothelial dystrophies are also genetic. But then your donor cornea will work because you don't have any problems in the epithelium because your vasculatures and everything is pet, and your donor cornea will support. So it is like a lifelong treatment procedure. And once your graft survives and it stably integrates, then you don't have a problem. So even if it is a genetic conditions in corneal endothelial dystrophies. So for other than that, I'm not aware of any other treatment procedures. For epithelial, and there are also laser-based method like LASIKs are done to correct because there's the unevenness in the cornea, so they interfere with the stromal layer to chop off some of the layer so that the layer cornea is made either flat or properly curved. So this is done by just laser ablation method. So I don't know again whether it is a stable procedure. It can also lead to fibrosis in case of reactive individuals. So it can actually worsen the situation. But then it is a clinically approved therapy that's been offered routinely in the clinics. So this is what I am aware of for cornea. And for the lens, again, unlike cornea, like endothelium, lens epithelium also doesn't regenerate. So they terminally maturate, differentiate and forms a lens fibers. But then there is a single line of epithelium which is there on the anterior margin, which stays throughout the life. But there are people, the recent studies that have shown that when you do a focal emulsification and remove the cataractous part of the lens and you leave the lens sac intact, these remaining epithelium can grow and regenerate to make the complete lens. There's one nature study that has come up. And there is also in vitro studies wherein the people have used some chemicals to break, because these are misfolded proteins that are there inside the lens bag. So if you can unfold them, and you can regain the transparency of the lens. So that is what has been done. But then stem cell therapy is not viewed here because you have a simple solution. You have simple cataract surgery wherein your synthetic lens can be implanted and it can stay for life. So when you have a simple solution which is working and time tested, not many people are investing in a stem cell based approach or any other complex method for lens based defects. So that's my perception.
Yeah. Yeah, I have some knowledge about the mechanobiology perspective you're talking about as well.
Okay.
That doesn't seem to work so well. So I think one or two groups have tried reshaping the lens, and
Exactly.
and it doesn't really stick. So the kind of interventions that Indumati is talking about are probably the only ones at the moment that are available.
Okay. And I mean, and is there any active research going on in either in CBS or in the Ocular Research Center, which is about to study the retinal organoids, as Madam was telling about in the end, and to see how the mechanical stress or stimulus can really change the behavior of cells in those cases?
Yeah, definitely. My group is definitely looking at using iPSCs as model systems. And we generate not only epithelial sheets of retinal or corneal epithelial sheets, but also organoids in suspension cultures. But our lab is mainly focusing on retinal organoids at the moment, but we have also published a paper on corneal organoids. But we haven't gone into the details of mechanobiology or looking at, we only looked at the early development and never modeled with any disease conditions. So probably those are the future directions that we'll be looking at.
Okay, thanks a lot for answering those questions, and I would have a look at the publications. Thank you.
Great. So,
Thank you.
Danu, you have a question? Danu.
Dhanu Lakshmi, you may unmute yourself.
Oh. No.
Dhanu, okay, there seems to be some problem.
Okay. So someone asked about the involuntary movement of the eyeball, and they're asking whether that's because of defects in the optical, in the orbital.
Ocular muscles.
Yeah, ocular muscles, sorry.
Yeah,
Yeah.
that's right. Yeah.
Yeah.
It is. It is.
And so the involuntary movements are happening just because of random neural firing or something?
It could be both neuronal as well muscular.
Okay. 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 So we have, I think there are a few more questions which can be asked. Someone asked what the viral vector was that was used in gene therapy, and that was adeno-associated virus, I think.
That's AAV2.
Yeah,
Guys form. Yeah.
Okay. And Neha also asked whether the retinal trial that you were talking about, either the induction of the retinal pigmented epithelium cells or AAV, were they showing any positive results?
Yes. So the autologous RPE, that the trial initiated by the Riken Center, Center for Developmental Biology, Riken. So they have done on 2 patients, and they have reported a 1-year and 2-year follow-up studies in EGM. They've shown what is convincing is that these cells, though it is an autologous iPSC-derived cells, it didn't trigger any severe immunological reactions. There was also another trial by ESCs that was done in the US. It's a multicentric trial. It's an ES-derived RP cells transplanted as suspension subretinally in a small group of patients. And there again, they have reported that it doesn't elicit any immune response because it's a subretinal injections and subretinal space is immune privileged. But that need not be true in all cases, because in some of the dystrophic conditions, when the RPE layer is compromised, it's no longer immune privileged, because RPE is the one that ensures your immune privilege status.
Okay.
So if there is a compromise, then we really don't know unless more and more trial outcomes come out. But severe immunological response is not reported so far, and visual outcome, it's very marginal in those few patients where the outcomes were reported so far, but long-term outcomes are yet to be awaited.
Okay. So LV Prasad is not doing any of these clinical trials.
We are in the process of doing preclinical studies right now in animal experiments, and we are definitely in that direction.
Okay. Okay. I wanted to ask one more question, which is the kind of preclinical studies on CRISPR-Cas mediated repair of mutations. And I'm sure that's probably one direction where these studies are going as well, taking, say, limbal stem cells and repairing them and bringing them back.
Yeah.
Yeah.
Yeah. So for retina, so whether it is gene therapy or gene editing, in situ gene editing, I mean not in vitro gene editing.
Okay.
So the challenge here is your retina should be in a receptive condition, in the sense that the cell should be alive, that your vector that you're delivering should deliver the gene, and the cell should be able to function and start expressing the protein. But if the retina is damaged to in a severe form, then just doing gene therapy or a gene editing construct is not going to work. So therefore, choice of the patient is very crucial. And another challenge here is identifying the gene mutation, because every patient will have different mutation, and there are not just one or two genes, there are 60, 70 genes involved. So say, let me give an example of RP65. So if we screen about 300 patients, you get about 2 to 5 patients who are positive for RP65 mutation, who carry a mutation in RP65. So in order to identify those 5 patients, you have to really screen 100 and 120 patients to identify those who could really benefit. Out of the 5, many of them whose retina would have been already degenerated just by delivering the vector, and the vectors are so costlier that a common man, like in our country, like in most of the developing countries, will not be able to afford those. That's a challenge that is in front of us, that we have to figure out how to come up with a solution. In case of cell therapy, here we are aiming to deliver the whole cell. We are not bothered about the kind of mutation that is present. We are trying to deliver the whole cell, and provided the cell survives there and regains its function and can regenerate, then it brings about a positive change. So
Okay.
here you can skip the screening process. You're not worried about the gene or tailoring anything for the patient. So it is an allogenic therapy of a normal cell delivered to the patients. But there are other challenges associated with the cell therapy too, which we have to address. So
okay, fantastic.
there is one CRISPR-based gene therapy. It's already in trial. It's called EDIT 101. It's for a gene called CEP290, which causes a form of LCA. It's again a labor congenital amaurosis. But then this CEP290 mutations, again, it's not anywhere in the gene. This is one hotspot mutation that they have identified, which is a splice site mutation. So they have designed guides to alter the splicing so that the splicing is regained in these individuals. Because it's a hotspot mutation, number of patients who would benefit would be more. So this is already in the trial, and this is one commercial product that's under testing now.
Okay, okay. So I just want to ask one last question, which is about, I read maybe last week or the week before about the bionic eye. So it's a bionic eye that can replace a damaged eye or something like that. Do you have any perspective on that?
Yes, I had kept a slide, but later I removed it. So, yeah, so yeah, it's basically a camera-based system. You have a camera in the front, and digital signals are then transmitted to a chip, which is implanted behind your retina, and then that transmits the signal. So the kind of vision that you will get, again, it's not yet available in the country. You have a license issue. Even the RP65 vectors are not yet available in the country. Novartis is still negotiating with the government. So this product, what it can do is, it might have like it will help the mobility of the patient. It may not help a person in reading or doing many of the bright light dependent activities, but it can help in the mobility of an individual to some extent, probably. That's what I can say.
Okay, okay. So with that, thank you, and thank you for giving us all of your time in the Friday before the holiday. So thank you. That was a fantastic talk. I actually learned a lot. So thank you very much for joining us.
Thank you so much.
Thank you.
It's my pleasure.
Okay.
Thank you so much. The talk will be available on YouTube. So, please share that talk with as many people as possible. Thank you so much, and I'm closing the call now. Bye-bye.
Thank you. Bye-bye.